Okay, hi guys. So this is the second part about enzyme. In this part, we will learn and discuss about three things. The first one is the factors affecting enzyme reaction, and then we will continue with enzyme inhibitors, and the last part will be about immobilized enzyme. So in the first subtopic about the factors affecting enzyme reaction, we will learn how to describe and explain the effects of pH, temperature, enzyme concentration, and substrate concentration on the rate of enzyme reactions. So actually, you have studied some of this in your previous level in a GCSE, for example, but we'll just continue and learn a little bit more details about this. Okay, so the first factor is the temperature. So this is how the temperature affects enzyme activity. So if you see in this curve, yeah, I divided this curve into three regions, A, B, and C. And then if you are asked to describe the effect of temperature on enzyme activity, remember, when you describe, you only describe the shape of the curve. Mention the y and the x-axis and also quote some of the data if it's provided. So for example, at this region A, we can describe as when the temperature is low, the rate of reaction is also low. But as the temperature rise, the rate of reaction also rise or increase until it reach something over here, which is B, the optimum temperature. And then above optimum temperature, when the temperature rise higher, the rate of reaction will decrease. Okay, now we'll see the explanation of this. Okay, so the explanation is like this. When the temperature is low, yeah, you know that molecules have kinetic energy, right? So at the low temperature, both enzymes and substrate, they have low kinetic energy. Therefore, those molecules are moving only slowly. So because they move slowly, there will be less collisions between enzyme and substrate, and less enzyme-substrate complex will be formed. That's why there is lower reaction rate only. But as the temperature rise over here, yeah, so does the energy, kinetic energy of enzyme and substrate. Because they have more kinetic energy, they collide more frequently, and then they have more enzyme complex in the solution or in this mixture. So that's why the rate of reaction increase until it reach B, or the optimum temperature. So optimum temperature is the temperature at which the enzymes works best. So this is where the maximum rate of reaction is being reached. Now, what happened above optimum temperature? Why the rate of reaction slows down? So you remember that the enzyme is actually a globular protein that has tertiary folding, right? In tertiary folding, there are many bonds inside that hold the structure. So when the temperature rises too high, remember denaturation occur. So in this case, hydrogen bonds will break down first because they are the, you can say the weakest bond. So when the hydrogen bonds breaks down, it will change the shape of the whole enzyme. Therefore, it will also change the shape of the active site. So because of the active site doesn't have its original shape anymore, the substrate, of course, will not be able to bind perfectly into the active site. Therefore, there will be less enzyme complex being formed and the rate of reaction will slow down until if we increase the temperature and to a certain point, the substrate may no longer fits at all and there will be no reaction at all. Okay, so the next factor is about the pH. So the pH also affects enzyme activity. Every enzyme also have different optimum pH, similar to temperature, where every enzyme has different optimum temperature. Yeah. So I have two examples of enzymes here, pepsin and trypsin. They have different optimum pH. So as you can see, the shape of the curve is similar to temperature effect, but in this pH curve, you may notice that this is more symmetrical in shape. So if the temperature, where's temperature? Okay, this one, it is a little bit uh, unsymmetrical between the left side and the right side of optimum temperature, while the pH, it's more symmetrical. Okay, that's how you distinguish between the pH graph and the temperature graph. Okay, so this is how you explain what happened when the pH is above or below the optimum pH. So as you know, in chemistry, you might also learn about this, that pH is the concentration of hydrogen ions in a solution. So when there is high amount of hydrogen ions inside a solution, it will make the solution will have low pH or acidic. while there is less or too low hydrogen ions in the solution, it means that the solution will be more too basic or alkaline, or we can say high pH. So every enzyme has its own, again, bondings, right? And including ionic bonds, okay, which is the interaction between positive and negative charged amino acids or side chain. Now, when you increase or decrease the pH below or above the optimum temperature, it means you are changing the concentration of protons or hydrogen ions, the positive ions, or we can say positive charge in that solution. So what happened? Now, the interaction between the R groups will be also disrupted, in which ionic bonds will be broken down. So because of the ionic bonds are broken down in enzyme structure, it will make the tertiary folding uh, will be open, and then the 3D arrangement of the enzyme will change. Again, it will change the shape of the active site, and the substrate cannot fit into the active site. That's why above and below optimum pH, yeah, the reaction rate will be slowing down like that. Okay. Now, this is how the enzyme concentration affects the enzyme activity. Nah, so the graph will look linear like this. It's just so simple. Okay, so as the enzyme concentration increases, the rate of reaction also increases. Will it always be like this? Actually, not really. It will be linear as long as there are enough substrate. So how do we explain about this? So if the enzyme concentration is low, like at this area, yeah, when you have little amount of enzyme only, there will be only little amount of active site, of course, and because there are low amount of active site, there will be only less enzyme substrate complex being formed, right? But as we increase the enzyme concentration, there will be more substrate can bind onto the active site. Of course, rate of reaction will increase, and so on and so on and so on until the substrate are no longer available anymore. So as long as the substrate are still available, yeah, if you increase the amount of or the concentration of the enzyme, there will be always a place for the substrate to bind to, and there will be always reaction, and the reaction rate will be increasing all the time. But if you don't have substrate anymore, okay, if you don't have any more substrate, there will be no more substrate to bind with the enzyme, no matter how much you add the enzyme concentration. And later, at some point, yeah, the rate of reaction will be levels off or constant or even decrease. Okay. Now the last is the concentration of substrate. So this is how the substrate concentration affects the enzyme activity. Let's see. So the shape is like this. So how to describe this? So when the substrate concentration is low, the rate of reaction also low. Okay, as usual. And when the substrate concentration increases, the rate of reaction also increase. But it will reach a point where you add the substrate concentration for too much, the rate of reaction will be constant or stays constant, or we can say levels off. Okay, like this. So what is happening? So what happened here is when you have low amount of substrate, okay, low concentration of substrate, there are still many active sites free, okay, and there will be not much reaction because of course there's only a little bit of substrate, so of course you will also only produce a little amount of products, right? But as the substrate concentration increase, more substrate will bind onto the active site of enzymes, there will be more complexes formed, and there will be higher rate of reaction. But at some point, when you add too much substrate concentration, you will reach something called Vmax or maximum rate of reaction. It means the rate of reaction will not increase anymore no matter how much concentration of substrate you add, okay? Because this at this site, at this Vmax, yeah, all active sites of the enzyme will also already become saturated with substrate. So if we see the diagram, it's going to look like this. So when there's low substrate amount or low substrate concentration, there will be more or some free active sites. But as you increase the substrate concentration, the enzyme will work more, there will be more complexes formed, more products being formed. But as you add more and more and more substrate until all of the enzyme's active sites are filled or occupied by the substrate, there will be no more empty or available active site to bind to the substrate anymore. That's why this free substrate will be like queuing up, yeah, or waiting in line to be able to be changed into products by the enzyme. Like that. That's why we reach something called Vmax. Okay. So overall, these are the four graphs that we already discussed, learned, described, and explained. Okay, so you review again the shape, yeah, between the effect of pH, temperature, substrate, and enzyme concentration, and make sure you are able to distinguish between describing and explaining. Okay. Okay, on the second part here, we will see the effect of enzyme inhibitor on the rate of enzymes reaction. So the first one, what is inhibitor? So based on the name itself, inhibitor are the molecules that can inhibit the work of the enzyme. So when there is an inhibitor present in a solution containing enzyme and substrate, of course the enzyme will not be able to work perfectly or as effective as usual. So there are two types of inhibitors. The first one is called competitive inhibitor and the second one is called non-competitive inhibitor. So we will see how both of them affect the rate of enzyme reaction. So this is competitive inhibitor. Okay, based on the term itself, competitive. So what does this inhibitor do? So competitive inhibitor, they like to compete with the substrate. Yeah. So this kind of inhibitor, they like to bind on the active site of the enzyme. So they prevent the substrate to bind into the active site as usual. So as the name, you know, competitive, they compete. Who comes first? They bind first, right, between the substrate and the inhibitor. So inhibitor that is competitive, they can do this because they usually have similar shape to the substrate. That's why they can stick and bind to the active site of the enzyme, but because it's not the substrate who bound to the enzyme, yeah, therefore there will be no reaction, no product at all. While the substrate, because they are not able to bind into the active site, yeah, they will still become substrate. They will not be changed into product. Okay, now the effect of these competitive inhibitors are usually reversible. Reversible, it means we can reduce the effect of this competitive inhibitor. How? So we can add more substrate. Yeah, it's just as simple as that because this competitive inhibitor, they work by competing with the substrate, right? Who has more collision with the enzyme, they will have more reaction with the enzyme, like that. So if we have only low amount of substrate concentration, with the presence of competitive inhibitor, of course, it will reduce the rate of reaction of the enzyme. So here in the curve, I have two types of line, the green one and the red one. The green one is without the presence of competitive inhibitor. Yeah, as you can see, as the substrate concentration increase, the rate of reaction also increase until it reaches the Vmax. But with the presence of competitive inhibitor, yeah, when the substrate concentration is low, okay, or not too high, the rate of reaction will be only low or lower than normal, okay, because there will be less substrate can bind onto the active site. But as we increase the substrate concentration, see what happened, we can also reach the same maximum rate of reaction or Vmax, just like the uh, absence of inhibitor, like that. Because at this site, yeah, we have enough substrate to compete with the inhibitor. So there will be more enzyme substrate complex being formed rather than enzyme inhibitor complex form. So this one is reversible, yeah. So if you have a competitive inhibitor presence in the solution of enzyme and substrate, just add the concentration of the substrate. It will reduce the effect of the competitive inhibitor. Okay. Now this is the example. Okay, for example, there's something or chemical called ethylene glycol, which is usually used in antifreeze, and sometimes this liquid is drunk accidentally. Well, I don't know how and why. So what is dangerous about this? So ethylene glycol, yeah, in our body, it will be metabolized into oxalic acid. So this oxalic acid is toxic. It may cause kidney damage. So because this reaction, yeah, is happening inside the body, of course, there is the enzyme that can catalyze the change of ethylene glycol into oxalic acid inside our body. So what we are going to do to reduce this effect is we just give something that can act on this enzyme, which is the inhibitor of this enzyme. So what is the inhibitor? It's ethanol or alcohol. Yeah, so ethanol, they can act as competitive inhibitor to the enzyme that catalyzes this conversion. Yeah, so when there is the presence of ethanol, ethanol will bind onto the active site of the enzyme. Therefore, ethylene glycol cannot bind to the enzyme, and there will be no oxalic acid produced until all the ethylene glycol is being removed from your body completely, like that. Okay, don't try this at home anyway. So the second one is the non-competitive inhibitor. Nah, non-competitive inhibitor is the opposite of competitive inhibitor. So when the competitive inhibitor binds onto the active site, the non-competitive inhibitor, they bind to another site other than active site. So it's binding to at any site elsewhere on the enzyme, but not on the active site. We usually call this site allosteric site. Okay, so this enzyme, yeah, this enzyme, okay, for example, they have active site over here and allosteric site over here. So when there is the presence of non-competitive inhibitor, this non-competitive inhibitor, for example, it binds to the allosteric site of this enzyme. See what happens, because enzyme is a protein and it's flexible. The presence of this non-competitive inhibitor will change the whole shape of the enzyme, and so that the active site shape also changes. So because the active site shape is changing, substrate cannot bind. Yeah, so because substrate cannot bind to the active site, there will be no reaction, of course. And in this case, yeah, adding substrate has no effect to this, because the one that is disrupted is the active site shape. Okay, like that. So no matter how much substrate you add, there will be no effect on the rate of reaction. It will be slowed down whatsoever. Okay, now this is the curve. Okay, the graph. The graph looks like this. So when there is no inhibitor, as the substrate concentration increase, as you can see, the rate of reaction also increase until it reaches Vmax again. But with the presence of non-competitive inhibitor, the rate of reaction will always be lower than the when there is no inhibitor, like this, and they will not reach the same Vmax as normal. Okay, because adding substrate concentration will have no effect on the rate of reaction. Now, this type of non-competitive inhibitor, it can be non-reversible, it can be reversible. It depends on the type of non-competitive inhibitor. And this non-competitive inhibitor is not always dangerous. Yeah, sometimes it's also useful. Usually that's the reversible one. So what are the examples? Okay, this is the example of the reversible non-competitive inhibitor that is usually useful in our body, and usually it is used in the metabolic pathway inside the body. What is metabolic pathway? Metabolic pathway is the reaction, yeah, that is happening uh, from the first initial substrate to make the end product. It will need series of reaction like this. So for example, from this first substrate to becoming the last product or the red one, we need 5 enzyme catalysis reaction A, B, C, D, and E. So this is metabolic pathway. It also happens inside your body, where, for example, when you digest or you eat starch, okay, the starch will not be able to be directly metabolized or hydrolyzed into glucose, right? You need several types of enzyme to make this reaction possible. For example, thiolin first or amylose inside your mouth, make it become maltose, and then later inside your guts, the maltose will be hydrolyzed further into glucose, like that. So this is like a chain reaction, and usually this metabolic pathway is always happening inside our cell. It's not only a simple reaction from A to B like that and then done, finish. Okay, so that is about metabolic pathway. So what is the role of non-competitive inhibitor in this metabolic pathway? Okay, so you will see this video to understand how the non-competitive inhibitor might uh, be like advantageous for us. Yeah, so that is the example of reversible non-competitive inhibitor that might be useful inside our cell. While on the other hand, there are also irreversible non-competitive inhibitor that might be dangerous. For example, this cyanide. So cyanide is example of the irreversible inhibitor, non-competitive inhibitor to one of our respiratory enzyme. So when you do respiration inside your cell, you need lots of enzyme. One of them is called cytochrome C oxidase. So when the cytochrome C oxidase is irreversibly inhibited by the cyanide, your cell will not be able to produce ATP as usual, and it may lead to cell death. Okay, so cyanide here is usually used for pest control, maybe for making pesticide. Yeah, so it is how they kill the pests by uh, poisoning and inhibiting the respiratory enzyme of the pest. Okay, so the last part will be about immobilized enzyme. So later we'll see what is immobilized enzyme, how to make it, and you will have to be able to explain the advantages of using immobilized enzyme. Now, but first of all, if you see this picture, okay, the left side is the lactose-free milk and the right side is the clear fruit juice. Yeah, you have learned before, right, about this, how to make lactose-free milk. We can use lactase or the enzyme that can digest lactose. So the milk produced is already free with lactose from lactose. While here, the juice, yeah, the fruit juice that they usually produce are already clear because they add enzyme called pectinase. Now, so remember that pectinase digests the cell wall components of the fruit flesh. So when you make it into juice, the fruit juice that is produced will be clearer and purified. Yeah, like that. So how do they make these two products? So do they mix the enzyme with the milk, or do they stir or mix the enzyme together with the fruit juice? Now, if it's like that, it will be such a waste for the factory because they will always have to uh, buy or add more and more and more enzyme to every batch of products that they produce, right? And then also it will be very difficult for them to purify the product because when you have milk already mixed with the lactase and then the consumer only wants the milk without the lactase, right? So it will be very difficult for them to purify the product and separate the enzyme like that. So therefore, they have something, or we usually use something called immobilized enzyme. So what is immobilized enzyme? Immobilized enzymes are the enzymes that have been fixed to a surface or within a bead or agar gel. So the enzyme is kind of like inserted into the agar gel. Yeah, kind of like boba if you see the picture over here, right? So if you imagine boba, inside the boba there are the enzymes there, okay? So how to make it? So the enzymes solution, yeah, this is the enzyme solution. It is mixed with sodium alginate solution. So sodium alginate is giving out like a gel-like structure. So when the enzyme is mixed with sodium alginate solution, then the droplets of this mixture are added into the calcium chloride solution. So this is a calcium chloride solution with syringe. Okay, the droplets of this mixture is dropped with syringe one by one. So when this mixture are put into calcium chloride, this calcium chloride will harden the gel. So the gel and with the enzyme trapped inside will be look like this. Okay, so this is the gel, alginate gel containing the enzyme like that. So this is how they produce the milk. So the beads or the immobilized enzyme inside the alginate beads are put into the funnel like this, okay? And then the milk that is still containing lactose will be poured from the top. And then when the milk have contact with the alginate bead here, of course the milk or the lactase inside the milk will be hydrolyzed into glucose and galactose. So at the end here, we will have the pure product or the lactose-free milk. Yeah, because the lactose has been hydrolyzed by the lactase here, like that. Okay. Now over here, we will have only the milk without the enzyme. Why? Because the enzyme still remains inside the beads or the alginate gel over here. Okay. So what is the importance of using immobilized enzyme? What are the advantages? So the first one, we can reuse the enzyme because in these beads, the enzyme are kind of like not mixed with the products, and then the enzyme concentration or the volume of the enzyme remains the same. So this will keep the cost down. Okay, so the factory or the producer doesn't have to buy another batch of enzyme. They just reuse and reuse and reuse this immobilized enzyme until they are denatured completely. And then the second one, we can purify the products easily. Yeah, so there will be simpler downstream processing they don't need to separate the enzyme with the milk, or maybe doing some another process after the digestion process. Okay, so it will purify the products instantly. And then the third one, it will give the enzyme longer shelf life, so we can keep the enzyme for longer time because they are inside the beads, they are trapped here. They're kind of like protected from the change of temperature or pH change, so they are going to be more tolerant to temperature change and pH change. This stabilize the enzyme, more shelf life, like that. Okay, so here you can see the video of the example how the immobilized enzymes are made and being used and being tested. Okay, thank you. So this is the end of the second part of enzyme. Now do your assignment. Thank you.